kind invitation to invite me here to share with you some of our most recent observations and treatments or approach to manage cancer. I'm at Boston College, a Jesuit university in Chestnut Hill, Massachusetts. I am not at Boston University. Many people make that mistake. That's another university down on Commonwealth Avenue. Just wanted to make sure that distinction was clear. What I'm going to do is just go over some background information on cancer and where we stand in our attempt to manage this disorder. So here you see the data on dead people, people diagnosed and who have died or projected to die from cancer. These data come from the American Cancer Society. One of their main goals is to log the dead every year. They do other things, but predominantly tell us how many people are going to die or have already died. And you can see these are the numbers from 2013 to 2025. The 2025 numbers are the projected numbers will be dead by the end of the year. You can see a steady increase, number of cases from 2013. There's a percentage increase here. When we take the total numbers of deaths per year and divide that by 365, you can see that we have about 1,700 people in the United States dying every day. And if you divide that by 24 hours, 24, you get about 70 people per hour in the United States are dying from cancer. And people, what's going on? Why do we, it's a relentless number of people dying every year and every day. And what's really interesting is we hear every year how much more money is given to cancer research, okay? NIH, National Cancer Institute, we always boast about how much more money we spend on cancer. Pink ribbon campaigns, the Susan Komen breast cancer. Everybody's excited by how much new money we raise every year, and all we get is more dead people. So what the hell is going on here? We have made raising money for cancer an end in itself, rather than a means to an end. Success is determined by how much money we raise, not on whether or not we drop the death rate. And that's kind of unusual. Where is the accountability? No one seems to be concerned about, okay, well, I'm giving you all this money year in and year out, and all we do is get more dead people. And then they say, oh no, we're making tremendous progress. And this is their evidence for the 33% reduction in cancer deaths in the United States since 1991. See, look at the progress we're making. And they'll come on and say, we're making tremendous. If you'll notice, this was the beginning of the anti-smoking campaign. The progress we have made is predominantly in the area of prevention. It's not in the area of new drug development. Remember that, it tells you. So those red lines are how many people more would have died if we didn't stop the smoking. So we want to now talk about the origin. How do we get cancer? What's going on? Cancer is defined simply as dysregulated cell growth. That's what cancer is, dysregulated cell growth. Caused by genetic mutations. I should say, is it caused by genetic mutations, or is it caused by chronic insufficient respiration coupled to compensatory fermentation, which I will break down? So you all know, we're all on the same page. So we have to talk about scientific theories, because it's the theories of how we understand science that move fields forward. So a scientific theory is simply an attempt to explain the facts of nature. Reality is based on replicated facts, whereas the interpretation of the facts is based on credible theories. Credible theories move science forward, whereas flawed theories stall scientific progress. Let's look at some historical examples. This is, what I'm showing here is the heliocentric theory that was put out by Copernicus, Kepler, Galileo. Could explain better the movements of the celestial bodies in our solar system than could the geocentric theory of Aristotle and Claudius Ptolemy. Ptolemy and his group, they used very sophisticated mathematics to try to attempt to determine where planets would be at any given point during the year. This mathematics led to epicycles, deference, and equants. Very, very complicated mathematics. Copernicus was attempting to use the same mathematics to improve our understanding of the movement of the planets, but he made a remarkable change in thinking. Instead of keeping the Earth as the center of the solar system, he made the Earth simply another planet, putting the sun in the center of the solar system. And all of a sudden, all this complexity seemed to be much less, the system became much less complex. Kepler knew that it was not simple circles, they were ellipticals. And of course, Galileo chose to use the telescope to better plot the positions of the planet. He saw the moons of Jupiter, and things started to fall into place in our understanding of where we are in the solar system and in the universe. The guy on the bottom, if anybody doesn't remember who he was, that's Giordano Bruno. Bruno was a heretic in the Catholic Church for many different reasons, but he also subscribed heavily to the Copernican theory of the organization of the solar system. And unfortunately, that was too much for the church at that time to deal with, so they burned him alive in Plaza Fiori in Rome, and we have a statue to him in that plaza, and he has been recognized as a martyr for science. The germ theory of Louis Pasteur could explain better the origin of contagious diseases than could the bad air miasma theory of Galen, leading to the development of ways to stop vaccines and antibiotics and things like this. Major advance in our understanding. The Darwin-Wallace theory of evolution by natural selection could explain better the origin of species than could the theory of special creation, moving the field of biology into a serious discipline. The problem we have confronted with us today, can the mitochondrial metabolic theory explain better the origin and management of cancer than can the somatic mutation theory? And I consider this just as important as the previous theories that I just went over. Does cancer arise from nuclear somatic mutations or from chronic mitochondrial dysfunction with compensatory fermentation? So we have a picture here of a cell that you see in biology textbooks to give students an idea what the mammalian cell looks like. It has a membrane, it has a big nucleus, and it has these little bean-shaped organelles called mitochondria. That's what it looks like in a textbook, but when you look on here, that's what it would look like under a microscope where mitochondria have been identified with a tag. Mitochondria are like a spaghetti network inside cells. They are very active, they fuse, they together, they separate, they move around, and they respond to their environment in remarkable ways. Today, the current dogma in cancer is that cancer is a genetic disease. The dogma is an irrefutable truth, something that should no longer be questioned. And this paper, Hallmarks of Cancer, The Next Generation by Hanahan and Weinberg, is one of the most highly cited, if not the most cited paper in all of science. Over 75,000 citations to this paper. And the statement from the paper says, cancer cells carry the oncogenic and tumor suppressor mutations that define cancer as a genetic disease. And this is what we see in all biology textbooks, cell biology, biochemistry, and genetics. Cancer is a genetic disease. The U.S. National Cancer NCI, National Cancer Institute, solidification of the dogma. If you say up there, how does cancer develop? It says cancer is a genetic disease that is caused by changes in genes that control the way cells function. And down here it says even more, cancer is caused by certain changes to genes. So there's no discussion about anything else other than cancer is a genetic disease. And most people in the world, when asked on the street, do you know anything about cancer? Yeah, it's a genetic disease. Well, let's look at the evidence that challenges that particular viewpoint. Listen, whenever you try to challenge a dogma, it's pretty much ignored. People don't want to look at it, they don't want to talk about it, they don't want to hear about it, they just simply ignore it. It's too controversial and disturbing to consider something that you have solidified in your brain as being the truth. And that's what I said here. Ideological dogma and confirmation bias prevent rational thinking. We are a species who are supposed to think rationally. But I'll tell you one thing. Ideological dogma is one of the most powerful forces that we know of in our existence. So now let's go through the evidence that kind of doesn't make sense according to the dogma. Now, I have references here, and I don't want you to simply listen to what I have to say, because you heard it from me. You should read the papers yourself. You're all cognitively capable of doing this. Most people don't read the original research articles, they rely. What do you think? What do you think? Oh, yeah, okay. No, read the damn papers yourself and make your own decision. So we have a number of studies that have been done where they can't find any mutations. Not many, but they're there. And every one of those papers that do all these studies and they find one sample or a few samples that don't have any mutations, they ignore them. They don't discuss them. It's unbelievable. I have the papers here. Please read them. You'll find in those papers that there are some cancers they found that, we didn't look hard enough. The search still continues. So you can't prove anything by negative data. So what the field did as we began our revolution in technology is we separated certain genes. Some are passengers that just kind of hang around and don't do anything. But the real bad ones that cause dysregulated cell growth are called driver genes. Oh, wow, okay, we got drivers now. Drivers and passengers and all this kind of stuff. And then we said, yeah, that makes a lot of sense, until our technology improved even further. And we were able to show that we have a lot of driver gene mutations in normal tissues. P53, that loving mutation everybody talks about. Pathological mutations are found in normal cells of people with P53, no dysregulated cell growth. Wow, no, that's a little harder to explain. Now we have these driver genes in normal tissues that don't form dysregulated cell growth. Wow, no, so what do? The search still must continue. Maybe there's something beyond the driver gene in the dark matter of the genome. Oh. Okay, let's start looking at dark matter. Just like the universe, we're looking at dark matter in the genome. The hunt for the ever elusive genes that cause cancer. Then we have some cancers that can be caused by carcinogens like asbestos that don't cause mutations. That's interesting. We have the rarity of cancer in our Aboriginal ancestors, and even to this day, we have tribes in Africa and certain remote areas that follow their traditional ways, and cancer is very rare in these people. How do we know? Because Albert Schweitzer checked it out, the great humanitarian physician, specifically looking for cancer. He said, wow, these Africans, man, they're really healthy. They have all kinds of parasites and other things, but they don't seem to have cancer. And other scientists in different parts of the world have made similar observations. And then we have the rarity of cancer in our closest biological relative, the chimpanzee, 98% similar to us in protein and gene sequences. There's never been a documented case of breast cancer in a female chimpanzee, and yet breast cancer is one of the leading killers of women in our societies. The nuclear mitochondrial transfer experiments, to me, are the most devastating and most powerful evidence against the gene somatic mutation theory of cancer. These are simple experiments that were done by Israel and Schaefer at University of Vermont some years ago. They had to scratch their head about the results that they got. Anyway, the green cell on top, a normal cell. This was an epithelial cell taken from a liver of a rat. It didn't look abnormal. They grew it in culture for many, many generations, and the red cell there looked abnormal in its way, its growth, and the way it looked. They said something's wrong with this, and they put it under the skin of a rat, and 12 out of 13 experiments showed a tumor developing from that cell. So the scientists said, oh, something must have happened when we cultured the original normal cell, or perhaps the original normal cell was mutant, we didn't know about it. So they went back and took the original normal cell and then put that cell under the skin of the rat, and they had no tumors out of 11 separate experiments. Then they said, let's try something different. Let's move the nucleus from the tumor cell into a normal cytoplasm, and let's move the nucleus of the normal cell into a tumor cytoplasm. And that's what you see. So when you take the red nucleus and put it into the normal cytoplasm, only 1 out of 72 independent experiments formed a tumor. Whereas on the other hand, when you took the normal nucleus and put it into the tumor cytoplasm, 66 out of 68 experiments gave us a tumor, so 97%. Scratch your head. What the hell is going on here? You should have gotten just the opposite results, not these results. So what do? Epigenetics. Oh, of course. With epigenetics, anything can be everything and its opposite. It's a very complicated field of genetics, and everybody has a different outcome when you'll start looking at it. But there wasn't only in vitro, and these are in vitro vivo experiments. We also have the work of Dr. McKinnell and his colleagues, where they were studying these kidney tumors in frogs, lethal kidney tumors. That's the frog with the big tumor there in his kidney. And amphibian embryology was, of course, well advanced. So McKinnell, I had a chance to speak with him before he passed away, talked a lot about these experiments with him. He wasn't interested in testing the somatic mutation theory. He was just asking whether the nucleus of a cancer cell could direct development. That was his thing. And then when I asked him, I said, did you realize that your results kind of challenged the somatic mutation theory? He goes, when you put it that way, yeah. So what he did was he took the nucleus out of the kidney tumor and put it into a enucleated fertilized egg, and lo and behold, they developed a tadpole, completely normal, developed from the nucleus of the kidney tumor. There was no abnormalities in that tadpole at all. And then when I asked him, he said, you know what I also did? I cut the tail off the damn tadpole, and they regrow their tail, they regenerate their tail. Perfectly normal, all driven by the nucleus of the tumor cell. He said the only problem with this experiment was the tadpole never could develop into a mature frog because there was something in the nucleus of the tumor cell that prevented complete development but did not cause dysregulated cell growth. Interesting. Now, Rudy Yanish and his group at MIT are the world lead. Rudy is one of the best in the world in mammalian developmental biology. So Rudy says, all that stuff that we saw there was other frogs and things like this. He said, I don't believe it until I actually do it myself. So he said, okay, he went back and he did it. He cloned a mouse from the nucleus of a melanoma. He took the nucleus of the melanoma, skin cancer, and put it into an embryonic stem cell. And there's the mouse that was cloned, that mouse there. I know it has gone through several stages of development. And then it, of course, aborted as well. But it never could complete the full process of development because there were a lot of chromosomal and mutational defects in the nucleus that prevented development, but did not cause dysregulated cell growth. These findings are inconsistent with what the somatic mutation theory tells us is the origin of cancer. So what I did is I put all this stuff together. I let a lot of other people do the work for me, right? All I had to do was look at the data and I said, I have a completely different interpretation of this. Because they were all interested in a different question. They were not challenging the somatic mutation theory of cancer. I'm looking at the results and I'm saying these data are lock solid against the somatic mutation theory. And they were repeated over and over again by the best developmental biologists in the world. So these weren't hacks that were doing these experiments, and they were publishing in the top journals. So of course I then summarized all this research in this diagram that many of you may have seen all over the internet. So normal cells beget normal cells. They have a great interactive genome. The genome looks great. The mitochondria are there in normal structure and function. And they have regulated growth, not dysregulated growth. Tumor cell, on the other hand, tumor cells beget tumor cells. They have genetic abnormalities, most of them in the nucleus, and they have abnormalities in the number, structure, and function of the mitochondria. And they have dysregulated growth. So the test was, of course, move the red nucleus into the green cytoplasm and see which should drive dysregulated cell growth doesn't. You get regulated growth. You saw that from the evidence I presented. You take the green nucleus of the normal cell and drop it into the cytoplasm of the malignant tumor cell, and you get either dead cells or cells with dysregulated cell growth. These are the exact opposite results that you would have expected if cancer were a genetic disease. How much more evidence do you need? Right? What the hell do you need? Oh, no. So what they say is it has to be repeated. What? Did you read the evidence? No, I didn't read it. It has to be repeated. One person said, top guy, if these experiments are right, the whole cancer field must be turned upside down. Therefore, they can't be right. And that is the truth. And I will not name the person who said that. If somatic mutations are not the origin of cancer, how do we get cancer? And that was shown many years ago by the distinguished scientist Otto Warburg from Germany. I don't have time to talk about his history. A lot of great books written about Otto. He was a half Jew. He knew that Hitler knew. He was spared during the Holocaust. He was spared during the Second World War because Hitler said if anybody's going to cure cancer on this planet, Otto Warburg would be the one doing it. So he let Warburg continue his work in Berlin during the war. Well, during the 20s, Otto said, all cancer cells arise from insufficiency of cellular respiration, which is oxidative phosphorylation. Energy through lactic acid fermentation compensates. If you're taking away one form of energy, you've got to compensate with another, which is a fermentation metabolism, which became aerobic fermentation. Some people call it the Warburg effect. I put this in red because this is our major contribution to the field. We cleaned up a lot of the mess and misunderstanding that Warburg was confronted with. We also found a second fuel that could be fermented along with lactic acid, and that is the amino acid glutamine. And unfortunately, Warburg nor the rest of the field actually understand that. They think glutamine is respired, it's actually fermented. We have a big paper that just came out showing that, which is the missing link in Warburg's central theory. Enhanced fermentation is the common pathophysiological malady of all major cancers. That is what we do know. What that means is that cancer cells are getting their energy from a different source than normal cells. This is our most recent paper, just came out, published in the Journal of Bioenergetics and Biomembranes, where I go through and show where everybody had made mistakes, the failed experiments, the missing control groups, and this kind of thing, cleaning up the mess for almost 100 years, allowing people to see the actual research and the experiments that were done and how we reinterpret them. And now it's open for people to reevaluate this whole cancer mess. Well, people say, are you sure the mitochondria are dysfunctional in cancer? Why don't we look at them under the electron microscope? Okay, you see that beautiful electron, those stripes, lines, those are called the cristae. They contain the proteins and the lipids of the electron transport chain, allowing us to get energy through oxidative phosphorylation. Here's the mitochondria of a glioblastoma, deadly brain cancer, and you can see they call it crystallisis. You don't see those stripes. The structure of that organelle is abnormal. Structure determines function, for crying out loud. If the structure of the organelle is abnormal, the function of the organelle is going to be abnormal. Well, that's glioblastoma. What about other cancers? Well, there's breast cancer. Nice cristae, vacuolated cristae. They're fermenting. Colorectal cancer. We have ghost mitochondria there. Structure determines function. If the structure of the organelle is abnormal, the function is going to be abnormal. Oh, but that's only... I went through every damn one of these major cancers, for crying out loud. It's not easy. You have to go back and read all these papers from all these age generations and stuff. And then you have to go through, none of these guys, they were just interested in what morphology looked like, morphologists. And in all of these major cancers, you find abnormalities in the number, structure, and function of mitochondria in all these major cancers. Okay? Blood cancer, every solid cancer. These represent 95% of all the deaths that we have are from these cancers here. And not only that, I went back and saw this. Turns out that all the major cancers collect big lipid drops in the cytoplasm. LD is a big lipid drop next to an abnormal mitochondria. I said, isn't that interesting? And then we went and saw lipid drops in colorectal cancer, blood cancers, breast cancers, all the different kinds of cancers. that have abnormalities in number, structure, and function of mitochondria store these big-ass lipid drops in the cytoplasm. You can see them. Turns out that when you have a defective mitochondria, they can't burn ketones or fatty acids. And in order to protect themselves, they store these as droplets in the cytoplasm. That's telling us that these cells are not able to use fatty acids for energy, because if they try to use the fatty acids for energy, you get reactive oxygen species and blow the damn cell up. So this is a protection mechanism. But 90% of the papers in my field say, because cancer is a genetic disease, those lipids are there to feed the mitochondria. How can that be? The damn cell, the mitochondria, defective in structure and function. Oh, I didn't know that. Jesus, you know? Anyway, this is the diagram from our most recent paper. You know, people say, how do you get cancer? Well, that was St. George's oncogenic paradox. Almost everything in the environment you know about can cause cancer, different amounts and different types. Carcinogens, yes, that's why we call them. They cause cancer or something. They damage the structure and function of the mitochondria. And now we have these forever chemicals, which are piled on top of all the other things we have in the environment. Intermittent hypoxia, radiation, age, rare germline mutations. We have a major paper coming out showing all those inherited cancers that you hear about, BRCA1 and all these others, Li-Fra-Many. They all damage in one way or another the structure and function of the mitochondria. Then we have oncogenic viruses. Yes, they go into the mitochondria, blow out oxidative phosphorylation, forcing the cell into a fermentation, dysregulates the growth. Inflammation and microplastics. And Faith Ann, little girl here, I don't know, she's here. She's going to give us a little talk on that. She becomes an expert now on microplastics and bubblegum, right? So she'll give you a lecture on that. I'll leave that to her discussion. We have oncogenes, and now we have what happens when you screw up the mitochondria. You get ROS, R-O-S, and that's the superoxide anion and the hydroxide that knocks the hell out of the genome, causes all the mutations, causes chromosomal defects. So you have to see genome instability, somatic mutations. Those are all downstream effects of damage to the oxidative phosphorylation. Will someone tell the National Cancer Institute that information? We're spending millions and millions of dollars hunting for gene mutations, all caused as downstream effects of oxidative disruption, oxidative phosphorylation. Now, when that happens, the cell starts to suffocate. Oncogenes, the HIF1 alpha, MYC, and all these others open the floodgates for fuels for fermentation energy. Once you stop oxidative phosphorylation, you must replace the energy, and we use these ancient pathways of fermentation facilitated by the action of oncogenes. So the oncogenes are facilitators. If you make normal mitochondria back in those cells, the oncogenes turn off. You don't need them. Oh, I'm going to go back one more. Here we got it. Now, how do we connect it to dysregulated cell growth? So we know we can get dysregulated because the mitochondria control the cell cycle through cyclins and the gradient in the inner membrane of the mitochondria. So cyclins, so when it's like liver, when they have to regenerate, when liver cells regenerate, they grow in a regulated way, okay? But hepatoma is a liver cancer that grows in a dysregulated way. So what's happening is that the liver regeneration is based on oxphos. Hepatoma is based on substrate level phosphorylation, which is fermentation. And because they acidify the microenvironment, crapping out the waste products, you get a lot of angiogenesis in there. People say, oh, mitochondria control the life and death of the cell, right? Yeah, they control apoptosis. They are the kill switch. If the kill switch doesn't work, cancer cells bypass the natural death pathways. Perfectly explainable. Apoptosis evasion, and I don't have time to talk about all of our years of work on metastasis. We know what metastatic cancer is. It's a fusion between a cancer stem cell and a macrophage in our body. One of the most powerful cells, and that spreads around your body. Because it's so glutamine dependent, we now know how to kill it. I don't have time to talk about that, but we can manage very easily metastatic cancers. So we have here on the bottom, the red line is the replacement of oxidative phosphorylation with the ancient pathway of substrate level phosphorylation. And you can see the greater the substrate level phosphorylation, the greater the malignancy. This is just as strong a relationship as gravity is to Einstein's redshift. Now let's break it down a little bit more. So we have normal metabolism. We know glucose is the fuel for making pyruvate through the 10-step Embden-Meyerhoff-Parnes pathway in the cytoplasm. Pyruvate goes into the mitochondria and is fully oxidized to CO2 and water. I'm breathing. Okay, get my oxygen in and I blow out CO2 and water. Those are the waste products of normal biological respiration. And you can see 90% of our energy. But you see we get small amounts of energy in the cytoplasm through the glycolytic pathway, and we get a little bit of energy through the Krebs cycle. That's called substrate level phosphorylation. You see that? Now let's look at the cancer cell. You see the huge shift? See, the energy now is coming out of these substrate level phosphorylation pathways. You get a bigger amount of total energy, much less coming out of oxidative phosphorylation. And when the cell does that, it loses its growth control. These cells are falling back on ancient pathways that existed before oxygen came into the atmosphere 2.5 billion years ago. So they're being fueled by these ancient pathways of substrate level phosphorylation. With the waste products of lactic acid and succinic acid, not CO2 and water, lactic acid and succinic acid. These cancer cells are crapping out those molecules in all the major cancers. The cancer cells have been telling us for decades what's wrong with them. We have just chosen to ignore it. Now, this is a real nice diagram put together by Derek Lee, my PhD student. Glucose and glutamine are the drivers of dysregulated cell growth in the cancer cells. And we can show how the carbons of glucose, the carbons of glutamine, and the nitrogen of glutamine can be so powerfully synergistic in driving the growth of these cancer cells. And we have put together all the major pathways, which I don't have time to go through each one of these, but you can see that there's a powerful synergy between using glucose and glutamine together. And what happens is they produce lactic acid and succinic acid, acidifying the microenvironment. People say, oh, you know, these cancer cells are so tough, they're so hard to kill. Immunotherapy didn't work, radiation didn't work, chemo didn't work, because they're protecting themselves with metabolic waste. What happens if you pull the plug on the two fuels driving the dysregulated cell growth and the production of waste? They're very vulnerable to death, easy to kill, not that smart. You hear all these crazy terms, very smart cell, very smart cell. Are you kidding me? Who would say something stupid like that? They have a growth advantage. Oh, I love the growth advantage. Over what? A cell that's not programmed to grow? A liver cell can outgrow a tumor cell when it has to grow. So if most cancer cells obtain energy through fermentation, what therapies might be effective for managing and preventing cancer? Well, one approach is to reduce the fuels that are driving the damn beast, right? So they need glucose. They need glucose. Glucose doesn't cause cancer. I don't know who the hell would say something like that. It's not good. The cancer cell loves to eat it, but it's not going to cause cancer. Okay, you got to lower the sugar, and you got to raise the fatty acids and ketone bodies. How do you do that? Water-only fasting, ketogenic diets, and this kind of stuff. Because I showed you the lipids are gorging the cytoplasm. They can't use those fuels. So the purpose of raising ketones and fatty acids is to let other cells in our body thrive. Because all of our other cells can burn either fatty acids or ketones, the brain, all these other organs, not the tumor cell. So let's get the person transition to nutritional ketosis first, and then go after their fermentable fuels, lower that glucose. And this is one of our earlier experiments on brain cancer in a mouse, which is a kind of a neural stem cell tumor. Neural stem, every here, oh, big A, neural stem cell. Yes, they grow very fast, but they don't metastasize. So metastatic cancer is more deadly than this. Anyway, AL is eating a high carbohydrate diet, unrestricted amounts, ad libitum. Give them as much carbohydrate mouse chow. Calorie restriction, CR. So what we simply did was we cut the calories down by 40% in these mice, and we shrank that tumor down. You can see by anywhere from 65 to 80% reduction in the size of the tumor. I said, wow, that's really powerful. I mean, all you had to do is pull the food away from the damn mouse and these tumors. So yeah, it seemed that. I said, wow, that's really interesting. So then we did it much deeper. We did a lot of work on this. This was way back in 03, 02. Each square is a mouse under a different dietary program. And we use linear regression. We didn't do correlation, we used linear regression. We chose glucose as the independent variable and beta-hydroxybutyrate and tumor weight as the dependent variables. As glucose goes down, ketones go up. It's an evolutionarily conserved adaptation. And in humans, it's even much, much bigger than what the mouse can do. As glucose goes down, the size of the tumor goes down. The higher the circulating glucose in the bloodstream, the faster the tumor grows. The lower the sugar, the slower it grows. Wow. Okay, we did this the first in the mouse. And then they said, let's see, does it happen in humans? Of course. We have dozens and dozens of papers in the scientific literature saying humans who have high blood sugar, if they have cancer, don't live as long as humans that have lower blood sugar. Innovate. Wow, geez, unbelievable. So if you want your tumor to grow fast, make sure you get your damn sugar up as high as you can get it. People like to race. Oh, your tumor can grow faster than mine. Then we built the glucose ketone index calculator. During this Q&A, I have stories about everything, right? This is, what do you think? I just pulled this stuff out of my butt? You know, this is, you know, oh yeah, wow, we have a problem here. Let's think of, can we figure out a way to handle this problem? So it would be better to measure combination. We had to take blood sugar millimolar, ketone millimolar, and make a ratio, because we found out it's not just lowering glucose, it's not just elevating ketones. You have to do both to allow the body to transition naturally into these new physiological states. So we made this ratio, and we found and predicted that ratios 2.0 and below are therapeutic for managing cancer, and not just brain cancer, all cancers. Then we built the press pulse, another long story about how we did that. Press pulse therapeutic strategies. You can press glucose down. We heard it's a non-necessary nutrient. We don't need glucose. Push it down as low as you can get it and run. Muscles will take and do a lot of different kinds of things. Keep your GKI as low as you can. Ketone supplements. Stress management, extremely important. People who are diagnosed with cancer, I'm going to die, you know. Raises corticosteroids, makes sugar high. We got to manage stress. You're not going to die if you know what to do. Then we pulse in drugs that will target glucose. We have to be very strategic about this. We just can't say one dose fits all. We are strategically targeting glutamine and glucose with small drugs while the patient is in nutritional ketosis. We move the person from a state of terminal condition, always stage 4 terminal, and we try to move him then into a state of management. We make the tumor much less angry. We have now time to work with this patient, to test things. Dosage, timing, and scheduling is where we are right now in the eventual management and control of this disease. Problem is, learning about dosage and timing and scheduling is not sexy science. Hard to get funding for that. But that's what's ultimately going to be what's going to happen. So, and we use hyperbaric oxygen as a replacement for radiation, because once you get that tumor cell into a metabolically compromised state, it becomes extremely vulnerable to high pressure oxygen. Now let's test this press pulse therapeutic strategy for a mouse model. We developed these models spontaneously. It was a spontaneous model, not these genetically engineered monstrosities that most of the scientific literature is filled with. 5 minutes? I just started. Nice telling me I got 5 minutes. I didn't take up the time in the beginning, my friend. Give me a break here. I come all the way from Boston. I got 5 minutes. 10 minutes.
Yay!
Why don't we do 15? I'll tell you what, you got to see the results, people. I'm just giving you the science. Because if I gave you the results without the science, you'd say, I don't believe it. Okay, listen, we stratified the mice. Let me speed it up. Let me speed it up here, for Christ's sake. Get me the hook here. Okay, so listen, we take a glutamine targeting drug, calorie restricted ketogenic diet, and I'm not going to go through all the details of the experiments, but you can see the blue line, the guy's just loving the high-carb food, died quick. Green line, a little bit of improvement. Orange line is the DON drug, a glutamine targeting drug. And when we put the two drugs together, the drug and the diet, you can see how much longer the animals live. We're really, really pushing extremes here. Okay, now this is human. Let's go talk about human glioblastoma. Deadly brain cancer. Deadly. You can see the midline shift. Most of these poor folks die from intracranial pressure. And the tumor cells, the purple cells on the histology slide, are cells running across the surface of the Virchow-Robin space, so you spread the tumor cells throughout the brain. That's why they're not surgically resectable completely. And this is the survival curve from 5 Canadian institutions. Look how replicable, look how beautiful the results are. The survival is so poor and replicated from one institution to the next. And this survival curve could be here in England, the United States, China, Japan, all over the world. You don't survive very long with glioblastoma. Why is that? And no improvement in 100 years. Are you kidding me? 100 years we haven't improved survival for glioblastoma? We have Webb telescopes around the Earth looking at the origin of the universe, and we haven't made any major advance in survival. And we found out that the treatment itself, when you treat a glioblast, you surgically resect and then you start the irradiation of the brain, and you break apart the glutamine-glutamate cycle, the brain starts to swell from the heat. You give the patient a lot of steroids, make the blood sugar go real high. You're feeding all of the fuels that the tumor cell needs to be very aggressive by the very treatment that you're giving to the patient. We tried this guy, he's Egyptian. Came in, he had glioblastoma, my colleague, this is from Alexandria, Egypt, young man, about 26 years old. His tumor was resected. We did a lot of good stuff on this guy. And then we got to irradiate him. I said, why do you want to irradiate him? The kid's really healthy. Oh, no, we must do it. We must do it. Now, we didn't have a choice. We must do it. So we did it. And then what happens, the guy goes back as a corn farmer, goes back out working in the fields, comes in, and he's got a headache. We published this paper, 24-month follow-up. Everybody was excited. Then he starts getting headaches. In 30 months, he's dead. Oh, what happened? So El Saka, first author here, said we're going to do an autopsy on this guy and see what happened. They didn't find any tumor cells. He died from radiation-induced liquefaction of the brain. I said, what the hell? I told you not to irradiate the guy. We have to do it. Now, this is Brittany Maynard. She young lady from California. She diagnosed GBM 2014. You can see her face. Here she is right after she got married. Look at her moon face is high-dose steroids. You get moon face from high-dose steroids. That means her blood sugars are through the roof. She says, I don't want to do this anymore. I'm going to take my own life, die with dignity. Wow. Then she writes an article in People magazine telling everybody why she doesn't want to go through standard of care and she's going to take her own life. It's not good when your patients kill themselves before they're going to take your treatment. Not good. Now, this guy is our English friend here, Pablo from Devon, England. He talked to Andrew Scarborough, who's here. Maybe we should do metabolic. Andrew was one of these guys who didn't want to do any stuff. He just said, no, I don't want to do that. He was no radiation, no chemo, no surgery. They said, you have an inoperable brain tumor. If you don't do radiation and chemo, you're going to be dead in 9 months. He said, I'll take my risk. So we put him on this I didn't know much at that time. He was like kind of our experimental case. He survived for a long time. He had IDH1 mutation. This is his brain tumor at the beginning. And then it grew very slowly, and he did a debulking surgery about 2 years after his first diagnosis. Now, why I published Pablo is because he kept meticulous records of his GKI, his glucose ketone index. So we have data to support why he lived so long. His GKIs were always down on the low. So I don't have time to tell you everything. But we published him as a case report to how powerful ketogenic metabolic therapy can be if done the right way, and Pablo was a pioneer in this field. Unfortunately, Pablo passed away immediately after the fourth debulking surgery of his previously inoperable brain tumor. Can you believe this? We spoke to him a week before he passed away. He had a cerebral hemorrhage in the last debulking surgery that ended up killing him, unfortunately. A good friend, Andrew and Pablo were very close. Now, this is another recent study that just came out of our group from Greece. They took 18 glioblastoma patients, and 12 of them didn't do, and it was a calorie-restricted Mediterranean diet. What? Salmon, sardines, olive oil, avocados, and that kind of stuff. And you can see the ones that stuck at 4 out of 6 survived beyond 3 years. doing this. And somebody said, wow, this is really remarkable. If we had a drug that did this, what did you do? I said, calorie-restricted Mediterranean diet. Oh, we're not interested. Absurd, right? Anyway, provocative question. Yes, ketogenic metabolic therapy should be the standard of care. This little boy's a tragic case, little Danny Sheen. He has what they call childhood glioblastoma. It's called pineoblastoma. And childhood, they go down the spinal cord. You get a lot of spinal cord involvement on this, right? So Danny, diagnosed 2017. You can see how fat his face is, massive doses of steroids, surgery, the same stuff, surgery, radiation, blood sugar high. Here he is a week before he passes away with his favorite Aquaman toy. A tragedy. What the hell are we doing to these kids? This is unconscionable. So I got so pissed off, I built my own metabolic pediatric model that replicated what goes on in Danny's brain and others. We had tumor cells going down the spine of the mice, so we replicated what goes on in high-grade pediatric, and we did. We have a movie. If you guys want to see how healthy these guys are on the right, look at the coat color. They're running around. They're enjoying life. They have restricted diet, MBZ. This is a parasite medication, and Don, and they live so much longer. And this is a case. We have Dr. Slocum here. He's here in the audience. You want to check out? We had this woman from Cleveland, Ohio, metastatic breast cancer to her bone, to her brain, all over the place. It lit up like a Christmas tree, for crying out loud. She flies to Dr. Slocum's clinic in Istanbul, and they worked on her, and you can see how this was a complete response. And he tells me she's still doing well. She's still doing well. He's here. You can talk to him. Look at her. She's happy. We did it with a lung cancer guy, non-small cell lung cancer. The guy's alive 10 years. It went to the brain, went to the bones, metabolic therapy. I did a puff piece for Nature, prostate cancer. You can manage that. This is our big paper that we just came out with. And I have those, Isabella's here, Matt's here, Abdul Slocum is here, Anthony is here, and Tom Durai, the first author, will be the leader of the field in the next several years. He and I published, and we have all of our colleagues, dietitians, surgeons, and all kinds of people on this paper. This is the clinical path to managing cancer, all cancers. I don't have time to talk about it, obviously, when we get the hook here. But anyway, it works on your dogs. Okay, we have a dog here, a pit bull. He had a big tumor on his nose, and she just put him on raw chicken. Oh my God. Raw chicken, raw eggs, fish oil, and the tumor just kind of melted away, and no surgery, no radiation, no chemo. The dog died from old age of heart disease. 15 and a half years, a pit bull, he lived much longer than the breed. So what we have, I don't consider these cancers stage 4 anymore. I think we're all terminal. Why do we have to make some people more terminal than others? We have a chance to keep people alive if they do metabolic therapy. So the answer here is yes. Mitochondrial theory can better explain the origin and management of cancer than can the somatic mutation theory. Please someone tell the National Cancer Institute that. I'm hoping RFK knows about that. Anyway, we have, let me conclude by it's a metabolic disease driven by mitochondrial substrate and cytoplasmic substrate. Press pulse is a therapeutic strategy. We are now working on strategies to manage all major chronic diseases. We're going to bring the hammer down on those things in no time. We're doing it now, and we're working on it now. So brace yourselves. Now, the other thing is, we started a new society, International Society of Metabolic Oncology. We're getting people, dietitians, nutritionists, surgeons, all kinds of renegade oncologists that want to join the society. Janet Nathan from Mumbai, India is our secretary. So we're putting this all together. And let me just say, I get all my money from private foundations and philanthropy. No gift to us is too small or too large. This is the way we work, okay? The more money we get, the faster we deal with the problem. So I want to thank all those who have supported our work, listed here, individuals and foundations, and thank you for your attention.
wanted to apologise for having to rush you and yeah, all of that, but I got to be the bad guy. I'm sorry, everyone. But no, thank you, Professor Seaford, absolutely excellent. But yeah, in the interest of time, award